Hello, everyone. My name is Armita. I'm at Max Planck Institute in Göttingen, Max Planck for Dynamics and Self-Organization. I think if you guys have talked to each other, you know some people from there here already, PhD students and postdocs. And I'm also at the University of Washington in Seattle. So it's a bit distant, but it seems that it's working. So I'm going to tell you a little bit about sort of using a statistical physics to study evolution and population genetics. Alex told you that basically sort of gave away in like how to use ways of thinking about evolution based on networks. And now I'm going to sort of build up a stochastic framework for studying population genetics. Before going into the science, I want to do an ad here, basically, because I think it would be relevant for a lot of you as students and postdocs. So we are organizing a meeting in Nordita. It's a school almost a month, coming July. And we're looking forward to get any of your applications because it's going to be very interactive school, both research talks and sort of background teaching for the students and postdocs who are interested in evolution. And the topic, as you can see, is predictability and control of evolution. If you have any questions, just write to me. But otherwise, you can just check the website and see what's there. All right. With that, let's move on to the science. When we talk about evolutionary biology, you can sort of think of biological forces that are involved. But for the purposes of this talk, we want to stick to minimal forces and biological effects to study. Let's just go over this cartoon of evolution. We all carry DNA sequences. They basically store information about function of an organism. So DNA has ACGT, and this is basically a code that would turn into amino acids and encode protein structures. This information basically translates to the functional features of organisms that then natural selection would act upon. So in this case, is a famous example of Darwin's finches. The different beak sizes have different efficacy in different environments, so they're selected based on that. It's kind of very much of a coarse-graining arrow here, but you get the gist of it. What happens is that these guys reproduce based on their features, right? Whoever is better in reproduction produces more offsprings, that their DNA is more similar to their parent. During this reproduction, there are mutations that are introduced in the genome. So that adds some randomness and some diversity in the population. So you sort of channel basically functional features here. They get slightly more diversified by mutation and other things like recombination and different biochemical changes that happen at the DNA replication level. And that gets back into sort of population configuration, right? It changes the whole configuration, and this circle goes on. And as a result, you end up having something like this, which is basically 4 billion years of evolution. OK, so in reality, it doesn't look like this, but yes, so this is a nice cartoon to think about it. But we're not going to explain this whole tree. What I'm going to build is sort of building a statistical framework to look at these. So look at evolutionary processes, to think about how we can use data to characterize evolutionary modes in a population. And I'll go between Blackboard and slides. And we'll see how far we can go. I have too many slides. Maybe I can't. Probably I can't cover all of them by the end of the talk. All right. So let's start with a simple toy model of evolution. Let's say we like spins because we're physicists. So our population has two types of spins, right? So individuals are defined by either upward or downward spins. And let's say we have a population and a fraction x of this population have green upward spin, and 1 minus x has sort of downward blue spins. And what happens is that, as I said, there are mutations that act upon these spins. They sort of change the direction. There's a thing that Alex also mentioned, reproductive stochasticity or genetic drift. That means if you have a finite population size and you sample from this population the parents that would reproduce, there's some randomness because there's randomness in how many kids you reproduce and how your genome is being propagated to the next generation. So that makes this process stochastic. So you sort of run evolution. You start from, let's say, frequency of X in a population to be small. There are mutations popping up. And at some point, you see that there is sort of the upward arrow is taking over. You go up to the frequency 1 for a while. That's what the configuration is about. And then you switch down, and you can go up and down. Now, if you add selection, meaning, let's say, the upward is beta for some reason, then the populations basically try to climb up a fitness landscape towards the higher values of fitness, right? So then at some point, you bias your dynamics towards getting more and more of an upward spin. And this climbing up is basically something like this. You can think of fitness as some potential that instead of going to the valley, you want to go to the peak. And you go sort of along the slopes. And you can define a selection coefficients, which are basically the gradient of this fitness function. And I'll be very specific about it in a minute. So this is just to give you a picture of what we're going to talk about. And so to build a structure, so let's write down sort of stochastic dynamics for these spin switches in a population. And that's simple, right? So we have now two types. Instead of frequencies, let's start with the numbers, because you'll see it's easier to sort of write down how these numbers change over time, and then we move to frequencies. Let's say we have two types, A and B, and A is the upward and MB is the downward, and let's try to write down how these ends change over time. Okay. So I would say I would define fitness as basically growth rate of a population. And if I want to write down how NA changes, so this is the exponential growth rate of the NA type with a rate equal to FA, right? That's the fitness. Then the mutations between the upward and downward, so you flip. So you go from the blue guys from B type to A type with rate mu B to A. And that should be proportional to NB, because these are the guys that actually flip. And then you mutate out of NA by A to B. And then there's a stochastic term that comes from this mating, like how many parents of NA are being sampled. And because we have these two states, it's basically just a binomial sampling from your population. And if you sort of go back to your stochastic processes that you learned, you can characterize this noise. So this is a Gaussian random number with mean 0. And also a variance. It's a delta correlated in time. And the amplitude of noise is basically a binomial sampling, so the amplitude is NA, right? So you can write down the exact same thing. Yeah?
I'm sorry, this term mu A to B and A. So the species A can just change into species B?
Yeah, so the mutations, they flip, you flip. So you're reproducing, and then you flip from A to B. Or
they reproduce.
Yeah.
But then the parent doesn't die.
Well, so this is, yeah, so basically you're keeping your population size, your population more or less constant, and that's what we say. In each generation, you select from the existing population, right? Some of them will be, so you randomly select N guys from your existing population, right? And there's a part that are NA, and these guys that are NA can, during reproduction, they can flip the spin direction, right? So then they become MB. Then if NA is a bit better than MB, right? So they grow as well. They reproduce with a rate that is proportional to FA, right? So that gives you this. So this is fitness. This is mutation 2. And this is mutation from. Right? So now, so it's not a birth-death process per se, right? It's just sampling, and then during each sampling process, you have mutation and selection, right? So it's a biased sampling of a population. You could write it as a birth-death process, and there is a mapping, exact mapping between the two. So this formulation is called, Langevin equation, right? Mathematically, it's a bit tricky because this is now you're writing some sort of deterministic derivative, and then you add some noise term. But there are ways of dealing with this. Yes.
Let me just ask a quick question.
Yeah.
So this is a binomial variance.
Yeah.
It should be PQ, right? So there is no MB in it.
This is for n. It's not for x. When you write it for x, the fraction, then you get it right. So I'll do the search in a minute, right? So you can write down the same thing for NB. So far, we are not assuming anything is constant, like n is constant in here, right? So you can just sample. But then we'll get there in a minute. And then you can write down the exact same thing for MB. But here, the fitness would be FB, NB. You get mu A to B and A minus mu B to A and B plus chi B. And to write it explicitly, so you can basically say there's no covariance between NA and NB, right? So if you put A or B, whatever it is, you get the noise amplitude proportional to its population size. All right. So we're interested in changes of the frequency, and frequency is x, frequency of NA, or upward arrows is x, which is NA divided by NA plus MB. And you all know your chain rules, so it's easy to go from one to another, right? And I'll just write down what the frequency of A type, how it changes over time, right? So this is the deterministic part. And now we have a stochastic part, chi x, yeah? Yeah, we'll get there, yeah. Let me characterize this chi x. So this is the noise in frequency, right? And you're absolutely right. If you have larger population, you should have less noise in frequency, yes? But yeah, so but it's a binomial, right? So the mean and the variance are equal, so Poisson process. Okay, so now let's characterize what chi x is. It's very similar to the deterministic part. Chi x would be chi b. And we can just write down what this turns out to be. Would be, so if I do the derivatives, you get exactly this form, 1 over x over n, which n is the sum of NA and MB, chi I plus minus x over n, chi b. Now, what we are actually interested in is statistics of the noise, right? And we want to have something like this for now chi x. So if I were to calculate what the mean of chi x is, that's easy. It's just the mean of this, which is 0. And And the covariance matrix, the variance of chi x, that we can calculate as well. And that's basically 1 minus x over n chi a of t times the same thing for time t prime and one thing we know is that there is zero covariance between A and B, so all the cross terms when I do the ensemble average will be 0. So the only things that remain are basically this term with itself and this term with itself. So it's the sum of the two noises. And you get 1 minus x square over n square chi a. We know what it is. It's NA. And then you get x square over n square MB. Then delta t minus t prime. And you put, NA over n is x, and b over n is 1 minus x. So you basically get x, 1 minus x divided by n, delta t minus t. So this is what your intuition was. The larger your n, the smaller your noise, right? So the larger your population size, the noise amplitude will become smaller. So in the infinite population size limit, we get basically a deterministic evolution. Also, it depends on x's, right? So the noise at the boundaries are basically, so when x is equal to 0, the noise amplitude 0. Is x equal to 1, the noise amplitude is also 0. So noise becomes larger in the middle. OK. So there are deterministic parts that you can put in. And once you do that, you basically get an evolution equation for x, which is equal to x 1 minus x times fa minus fb. So now I'm just doing a simplification, and I call mu, which is, I set the two mutation rates to be equal. You can keep them different, but we don't have much time here. So once you do that, this would be a Langevin equation for the frequency of the type upward arrow, right? Frequency of A. So as you can see, you could have guessed part of this. So what is this? This is the difference between fitness of A and B, Fa minus Fb. X1 minus x is the variance in a population with two types of A and B, right? So variance of the types. So this is variance. And you could do the exact same thing if I now take out the amplitude of the noise out of my chi. So you get x1 minus x over n chi x, which is now chi tilde x, which is now mean 0 variance 1. This is basically sampling from a population with variance x1 minus x. So standard deviation of the mean, if you wish, is proportional to 1 over n. OK. So this is the simplest sort of evolution equation you could write, and I'm going to extend this to higher dimensions and say how complicated it can become. But the formulation remains the same. So you have a variance here that sort of multiplies to your fitness. The larger your variance in a population, the faster it can evolve. Then that's somewhat important concept to keep in mind. And then sort of sampling from the population is also proportional to the noise, to this variance. So this is a Langevin formulation, and we know there is a mapping between Langevin equations and Fokker-Planck, and I can write down a Fokker-Planck formulation for this. That'll be of this shape, right? So the first term, the diffusion term, comes from the noise, and you see the amplitude is x1 minus x, so it's a multiplicative noise. It depends on your variable. And this distribution basically describes now a probability of observing a frequency value x, right? So now you run, so this was one population. Now you run parallel sort of evolutionary processes, and you ask, what's the probability of observing the upward arrow to have frequency x across this population? So this is a sort of ensemble description. And yeah, so the first term is a diffusion term. The second term is mutation term, and it's just what we have here. You put it over there. So it's a first derivative, second derivative, and the last term is a selection. And yeah, so I sort of also, again, emphasize the structure of this variance matrix. And you can solve this. This is called Kimura's equation. It's been known 68, I think, sometime around there. You can solve this. The solution. Yeah. You can solve this, and it becomes actually an interesting solution.
I think in practice you solve the backward equation.
You solve it backward equation, but you could solve it also. You could solve it forward. Yeah, you could solve it. You solved, when you want to calculate fixation probability, you solve it backward. Yep. All right, so you can solve this equation in the equilibrium solution for this. The time-dependent solution, I don't think there's a closed form for it. There's an equilibrium, a stationary state. It's one-dimensional stationary state and equilibrium are equal. So you can solve the stationary state of this. And all right, so the P0 has a structure. I sort of separate the two pieces. There's a neutral part and there's a part on the selection. If I put selection difference between the two types to 0, you still can solve this equation, and that's what I would call P0 of x. And that's a distribution that here is shown in black. So this is U-shaped distribution, and you could have guessed the shape of it, at least for small mutation rates. So okay, this, I think, I haven't written this down, so I think I sort of up to factors of 2. I think this should be something like that. Approximately. So am I having 2's right here or not for the neutral Kimura equation?
Close enough.
Close enough. Yeah. Approximately. Yeah. So you get this. Now if you crank up mu, what you end up getting it'll be actually a sort of bell shape. So you become sort of focused in the middle rather than having the largest value at the boundaries. So this is a regime of small mutation rate, meaning that you spend most of the time either at upward or downward. Now, if you make mutations larger and larger, then you become a random population. Half of you are up and half of you are down. Now, if you add selection, so there's a selection difference between up and down, you sort of tilt this distribution. It will be skewed. So in this case, let's say the upward is beneficial. So you have the majority of your population to be in the upward, so x around 1, right? And this comes in with this sort of Boltzmann type factor. You can build a lot of analogies between what we know from statistical physics and this formulation of the population genetics. So the sort of the relationship between neutral distribution and the selected distribution is the difference is this 2n e to the 2nf, and you can treat your 2n as 1 over kt and your f as an energy function. This is only analogy. This is not a physical system. We don't have a conservation of fitness as we have a conservation of energy in physics. This only comes from the sort of the structure of the stochastic process, and in particular the fact that we have the covariance matrix, or the variance in this case, showing up the same, having the same structure in front of your noise term and in front of your selection. That's why it comes out like this. Okay. This was basically a one-dimensional genome with two states, right? In reality, our genomes are slightly longer. In humans in particular, we have about 10 to the 10 base pairs. So if you were to think of all possible genotypes that you could construct, it's 4 to the power 10 to the 10. It's much larger than human population size. It's an interesting twist to it. So we can either write down a higher dimensional version of that equation, and I'm going to write it formally down, but then solving that equation, it's not very simple. Or we build up a different formalism, and that's where we're heading. But just to give you a picture, so this is a microscopic description of this process. You have a lot of genotypes. You look at all the mutations and all the positions, and you're living in a very high dimensional space. Traditionally, it was represented by this topographic map of fitness to sequence space, also drawn in 2 dimensions, which is misleading, but that goes to 1930s. These days, you could, in certain cases, measure the fitness, genotype fitness maps, and I think the last talk today, we will hear something about it. So this is basically a fitness landscape for a specific fungus that is sort of the experiments are done in the lab of Ariane de Visse. So in this case, you can see the genotypes are represented in a 5-dimensional space, basically 5 genes, and you would say whether they're Y type or the mutants. And you look at all the sort of transitions in this 5-dimensional hypercubes between all the states. So we can sort of try to better characterize this highly sort of rugged fitness landscapes in maybe a sort of certain specific cases we are interested in. But in general, this is a complicated problem to formulate. So, but formally, if we were to write it down, instead of having a single frequency, independent frequency value, which was x, because the other one was always 1 minus x, in this case, in k plus 1 dimensional space, right? So we have k independent frequencies that you can characterize, right? And ideally, you want to have a diffusion equation, a generalized diffusion equation, for change of this now frequency vector. And it'll look like this. And I'm keeping Einstein notation, so I'm not putting the sums. You sum over the upper and lower sort of indices that are in common. And there is analogies to general relativity at some point, if you wish. I'm not going into that, but interesting sort of mathematical features about the topology of this space. You can look into that. But for our purposes, let's try to understand at least what this function will look like in a 3 allele case rather than the 2, right? So to be specific, this G alpha beta of x is now a covariance matrix. And the F, capital F and capital M, are basically potentials, fitness potential and mutation potentials, that would then couple to this covariance matrix to tell you how you move in this manifold, given your slopes, right? And okay, I can save some of these things. All right, so if you have 2 types, right, so 3 types, we have XA plus XB plus XC equal 1, we have the covariance for at the level of populations, sizes of each type, the exact same thing as we had before. But what I'm going to do is that dx dt, for example, will have a third term, which is nc. So the term I'm going to calculate is the covariance matrix, because it looks like as if that once you know that, you can actually write down things very simply. So let's try to figure out how that looks like. So this will basically have a third term. And now I put XAs. See. Okay, so if I do the same sort of exercise as we did before, so this would give me the diagonal components of my covariance matrix. So the covariance between XA and XA fluctuations, right? And the same thing, there's delta, sort of delta function between A and B, so the unequal indices. So I basically get 1 minus XA squared over N and A plus XA squared N squared and B plus XA squared N squared and C delta in time. And you write this whole thing down, and what you end up getting is XA 1 minus XA divided by N delta. Now, we can also calculate off-diagonal terms. XB of T prime. And they're non-zero components because you can multiply the chi A terms here with the chi A terms here, but it comes with a different prefactor. So what you end up getting is basically minus XA XB divided by N delta T. So if I were to write my covariance matrix in this case, Gij is 1 over n. So the 1 over n comes, I'm actually putting it outside, so let's get rid of the 1 over n here. Would be xi 1 minus xi for I equal to j. And it's minus xi xj for I different than j. So off-diagonal and diagonal terms. And you can sort of try to convince yourself that this structure of covariance matrix applies to even higher dimensions than 3. And this is a special matrix. It's called a, Fisher, okay. This is a covariance matrix. You can make an inverse of this matrix, and it gives a metric on your space of frequencies, and that's called a Fisher metric. So the space is curved, and that's how it relates to what you know from GR. But it's a bit, yeah, so the distances, basically, you can calculate it like this, right? So the distances along this sort of frequency manifold, you can estimate it like this, and that would be basically telling you if I move a little bit in the frequency of this guy and the frequency of the other guy between 2 time points, how much I have moved in the frequency space during evolution. Right, so once you put this in, you can characterize your generalized diffusion equation for changes of the frequency in a population. Yeah?
When you had only two, like A and B, it was diagonal, but then now it's like this J, like the chi A, chi B, it was 0.
Where? No, it's not 0, right? So
I thought it was, you said that in the, like the first example you did, it was 0 for the odd diagonal term?
so it was only one term in the first example, right? So you have only one independent variable, which is x. And if you have 3 dimensions, you have 2 independent variables because they sum up to 1, right? And that's what it comes down, right? So once you go beyond 1 dimension, you get off-diagonal terms. Right. Okay, so building this, like this equation will be difficult to deal with. But the structure of covariance matrix is useful if you want to now map it or coarse grain your dynamics into what I will describe as a phenotypic evolution. All right, so what do I mean? So we start from the genotypic description. This was sort of a microscopic representation of your evolution. And then you can move to what is known as molecular phenotypes. So these are things that you can actually measure in the lab these days. So it's a lower dimensional phenotypes like binding affinity or gene expression values, things like that. They're encoded by many genetic loci. So if you were to write down an evolution equation at the genetic level, it will look like something that we wrote down in the previous slide. But the question is, can we do it at the phenotypic level in a way that it disentangles from your genotypic space? And once we do that, we would be able to then represent, so look at the dynamics of phenotypes. And we know that the fitness to phenotype map is often much smoother than the fitness to genotype maps. And that would then hint us how evolution would sort of factor in during dynamics in the phenotypic space. Let's start with a simple case. So far, everything has been in equilibrium, so time independent. So I will sort of be cautious about using the term equilibrium, because once we use high dimensions, you can actually build up curls, stationary probability curls in probability currents, and these things will stick around even in the phenotypic coarse graining. But the forces are time independent in a way that your selection or environment is time independent, and you can sort of try to do the coarse graining in this sense. So it's a stationary state, time independent dynamics. So let's do a simple mapping. In this case, let's say this is my genome, and the 2 states at different positions, but my phenotype is now a linear map between genotype to this quantity E. Your 0 or 1, right? Sigma I can be 0 and 1 depending on what you observe. But different positions have different contributions to this phenotype. And these are these quench variables, EIs, which would then tell you which ones are more important sort of in your energy estimate. So if you can define a scale for this phenotype, which is just sum of the EI squares. And that's kind of important to compare apples and oranges. So to compare height and, I don't know, weight, I suppose. So once we look at phenotypes, then the state of population, we can describe it as distribution of phenotype within a population. So let's say if we stick to law of large numbers, let's say things are Gaussian. And you can deviate from all these assumptions to some extent. So if things are Gaussian, there is a trait mean, so phenotype or trait average, gamma, which is just the mean of this phenotype distribution, and trait diversity, delta, which is the variance. And in principle, we are interested in how this whole distribution changes over time. Alternatively, we would be interested in how the moments of the distribution change over time. And in the Gaussian case, the only 2 that matter are the first and second. So we would want to characterize an ensemble probability density of the mean and variance of a phenotype population, a phenotype within a population, gamma and delta. So it should look something like this. And now the covariance matrix is not anymore over in the genotype space. It's defined in your phenotype space. Although we have 2 dimensions, but these 2 dimensions are not like frequencies that they sum up to 1. So you get 1 independent variable. Rather, you get a covariance matrix in the space of gamma and delta. And what we would like to do is to do a mapping that takes us from genotype space to the phenotype space. And that you can do. And let me start with sort of a specific example that then tells us what this mapping should be. So let's look at the case that we have a fitness landscape here that is quadratic. So this would be this term. And we want to know how your phenotypes or mean and variance changes depending on your fitness function.
Can you say a bit more about the random process here for this last equation?
So
it's not a single population, you say it's interpopulation.
So this is right, like before, right? So we had P of X.
Yeah.
So Q is basically ensembles of populations that have different gamma and delta values. And I'm going to get into what the G's are in a minute. Yeah.
Are there experimental evidence for seeing a formula between phenotype and genotype?
In certain phenotypes, yes. Right. So like binding affinity to some extent is a linear map between sequence and binding affinity. That doesn't mean your fitness is a linear function of binding affinity, right? Okay, and so that's the fitness, how the fitness of the organism depends on the gamma and delta. Okay. So I'm gonna sort of, what I'll do is the following. I'll try to write down a Langevin description of this dynamics for the mean, and then refer you to the variance. So basically give you a way how to calculate these things. So let's say I start from the selection. That's the easiest part. And so intrapopulation, so within population description of the phenotype, so let's call it W of E. This is that gray distribution, right? So phenotype distribution within a population. If I were to calculate what the gamma, the mean phenotype, how it changes over time with selection, you get this description, right? So gamma is basically, in the continuous limit, is the integral of E, W of E, dE, right? And that's basically equal to and that's basically asking how your density of a specific density or probability density for a specific phenotype changes. So if I say my phenotype, my fitness of a strain alpha, it basically depends on its phenotype E, f of E alpha, right? So I can then write down what this change in the density function is, which is just the integral of E. So what is ddt of W? It's basically f of E minus the mean fitness in a population, W of E. So this is very similar to what we had before, right? So we had how the frequency of a certain genotype changes, and it was proportional to fitness minus the mean fitness, or the fitness difference between the populations. And the fact that you subtract the mean fitness is to keep things normalized. So that's the only thing. Now we can sort of write these things slightly more explicit. If now I was arguing I want to write my fitness function to be quadratic, so let's call it minus C0, E minus E star square, then I can solve this equation, right? I can explicitly write this down. The average fitness, which is the average of F alpha, is equal to minus C0, the average of E minus E star square. And things are Gaussian, so this will be equal to minus C0 gamma minus E star square minus C0 times delta. So this is the deviation of the mean population from the optimum, and that's the variance of the population in this case, right? So because it's Gaussian, you get this formula. So that's your mean fitness. And you insert it here. You look at the Fe minus F, and you do the integral. You put the e's. And since things are Gaussian, you basically get the following. Minus 2C0 times delta gamma minus E star. So this was the change in the mean phenotype due to selection. And as you can see, there are things that you kind of know, right? So let me take this minus 2,0 times delta. I took the delta out. So this is basically the derivative of this F, mean fitness, with respect to gamma. And that's what comes out over there. So d gamma of F. And this is your variance, your interpopulation variance. Yeah?
Can you explain again, please, how you go from the derivative of W of E to that being equal to F of E minus E over?
Yeah, so you remember how we defined fitness for the genotypes, right? So you would say the change in the number of upward arrow was equal to the fitness of that times that number of the upward arrows. Then we move to the frequency of the things in upward direction, right? And then the equation was Fa minus Fb, so d dt of x is equal Fa minus Fb times x. And if you sort of think a bit more about what this Fa and minus Fb is basically Fa minus the mean fitness of the x's, which is 1 is Fa times x plus 1 minus x times Fb. That would be the mean fitness in your population in this case. Yeah, so you get Fa minus Fb times, right. Exactly. So the idea is you want to keep the frequencies to sum up to 1. That's why you subtract mean fitness. In this case, you want to keep your density to be normalized, right? So you're going from, a binary case to a continuous distribution, and you subtract the mean fitness so that your W remains normalized. So that's the only sort of trick. All right. So this is sort of a hint that my covariance matrix for gamma should be delta. You can derive this sort of properly, but we saw the hint because it should appear over there. Like G alpha alpha in this case would be G gamma gamma should be equal to delta. The other hint for that is basically these things are fluctuations of your mean. And we know from statistics, if you have a distribution, a fluctuation of the mean due to sampling is variance over the number of samples you have, right? So this is the variance, and now you get 1 over n because of the number of individuals, number of samples that you have that describes your empirical distribution. You get delta over n. But these are all hand waving. The proper way of doing this is to go back and do exactly what we did as before when we did the mapping. So to compute G gamma gamma, which is equal to now alpha and beta in the gamma and delta space. Actually, sorry. So these alpha and beta are genotypes. Because we know what the genotype noises should be. Now, so, okay, so this is a bad notation. Let's call this guy G tilde. So this is the phenotype. This is the genotype. So this is the thing we wrote down here, right? So if alpha and beta are equal to each other, you basically get x alpha 1 minus x alpha. If they're not equal, you get minus x alpha x beta, right? And we know how this relates. We know how gamma relates to x alpha. Gamma is basically sum of E alpha x alpha. It's just the average of E in your population genotypes. So you basically get the following. So this is E alpha, E beta, G alpha beta. And you can open this thing, and you basically get some. So in case alpha and beta are equal, you get E alpha square, x alpha 1 minus x alpha. And if they're not equal, you get minus sign E alpha, E beta, x alpha, x beta with a minus sign here. And you sort of move around terms, and what you end up getting out of this is E alpha, sorry, basically you basically get E square minus average E square, which is equal to your delta. So that's just a simple exercise. You can do the exact same thing for delta, and I'm not going to write it down. What you end up having is the fourth moment of your distribution, generally speaking, minus the delta square, which in the case of Gaussian would be basically equal to 2 delta square. And for the off-diagonal terms, so G gamma delta would be M3, the third moment, which is equal to 0 in Gaussian.
And the fourth moment is the case where it can factorize into a product of second moment?
That's the Gaussian, right?
Oh, you just wrote that down.
Yeah. All right, so you basically get a diagonal matrix in this case that your G tilde is equal to delta and 2 delta square. 0, 0. And that's this delta when you put beta and alpha equal to gamma. Okay. You can try to write down what the mutation term is, and I'm going to sort of skip over that because I want to cover other things, but it's the simplest term actually in the whole thing. So we got the selection component, we got the noise component, and I'll tell you that would be a mutation component, right? So if you write down basically a Langevin form for the mean phenotype, you get d gamma dt is some sort of, it looks like an Austen-Uhlenbeck process, right? So there's a mutation term that has equilibration towards some random state of your genotype space, and there's a selection component that wants to push you towards the optimal phenotype E star, and there's a noise with the amplitude delta over n. You write the same thing for delta, and you get a noise that is proportional to delta because your variance is delta square. So that's a bit tricky. It basically means that you have coherent fluctuations in your system. So very long tail distributions as a result. So the larger your value of variance, the larger are your fluctuations of variance, and that we know what is always the case for variance. I'm going to write down, I don't have it on the slide unfortunately, I'm going to write down a projection from the Langevin to a two-dimensional Fokker-Planck in this case. And it'll look like this. Delta. 2 delta square. So these are the diffusion terms. And then the mutation terms look like this, 2 mu And this is a term that is interesting. It comes from Ito calculus because your variance is a second order term. So write it down and get this term. It'll be satisfactory, I would suppose. And then you have a selection, right? So 2C0 delta, the derivative with respect to gamma, gamma minus E star. And if you were to guess what the selection term for variance should be, it should be basically the noise term, and then the derivative of mean fitness with respect to variance. The derivative of mean fitness with respect to variance is minus C0. That's our mean fitness. So you put it here, it'll be plus, and you go from Langevin to Fokker-Planck, so you get the C0. And then you get the first derivative with respect to delta times this variance term, 2 delta square. Okay. So diffusion, mutation, or sorry, genetic drift, so to speak, mutation, selection. So in principle, this is a two-dimensional Fokker-Planck equation. You can show that this is not there's no equilibrium solution to this. There's always so even in the absence of selection, you always have curls in your probability currents. But what comes out of this is that, as I said, fluctuations of delta are coherent. You have large fluctuations. And as a result, delta will become a very fast variable in your dynamics. So what you can do, you can sort of look at separation of timescales, and that's a useful thing in physics generally. So you can treat delta as a fast variable and just insert the average delta in this equation, you're reducing the whole dynamics to a one-dimensional diffusion of gamma. Alternatively, you can treat gamma as a constant and look at the dynamics of delta if you're interested in shorter time scales. If you do scaling, you can write down, you can rescale your variables, and you end up having this kind of rescaling. Your gamma fluctuations follow law of large numbers. Your delta is coherent. It scales with L. And if you do the thing I said, you sort of look at slow variables like gamma, you can build up some marginal distribution of gamma that you sort of trace out basically delta from this dynamics, and you get an equilibrium distribution. Again, it looks like a Boltzmann type distribution that takes the solution for the neutral part and multiplies it by e to the 2n now f gamma, which is this guy. And
where do we get your scaling? Often it comes from a conservation law. Right?
Yeah,
So
there is no conservation law. Yeah, yeah. So it's just you do a scaling with the genome length, basically. That's what you end up getting, right? So L is a genome length. And so in the long-term limit, so in the limit for that you're interested in dynamics of the mean gamma, you get a characteristic time scale, which is 1 over the curvature of your fitness function. And that's the C. So that would be your equilibration time. If, as I said, so you can write down these equations in a unscaled form. So these are the equations of gamma and delta, the equilibrium solutions for gamma and delta. The first one is a Gaussian distribution. The black part is a neutral term here. And this is your e to the 2nf. So the selection term, and this is basically what happens if you, so this is, this was your neutral evolution, you turn on selection, you move your mean towards closer to E star, around 70, and you move your distribution. As a result, your distribution will have a smaller variance, and it also moves to your optimal value. So this is for delta. Now you can see the structure of the delta or diversity. The distribution is by no means a Gaussian distribution. It has very long tail. That's where the coherent fluctuation comes from. And so if you turn on, so this is the empty circles or the neutral case, the black terms here. And you turn on selection, and you're basically cutting off the long term, exponentially cutting off the long tails of this distribution. And that's where selection comes in. So what are these things good for? Let's try to build up some observables from these statistics. And the first observable that you can actually measure from data would be to try and look at time-resolved phenotype divergence. So let's say I measure my gene expression at one time point in a population, and I measure it again at time point t plus tau, and I do the sort of deviation of this mean phenotype to the power 2, and I look at the expectation value of that, and I call that a divergence, a time-resolved divergence. You can make this dimensionless by dividing divergence by the diversity, and that's always good to get rid of, you know, apples or oranges in your problems. And there is sort of a quantity here that kind of takes care of how much genetic diversity we have, so let's not get into that. But you can construct this quantity that from theory we would know this will saturate to value. So this is a function of tau, time lag. This will be equal to 1 at tau goes to infinity for neutrality. Now, if you crank up selection, you increase selection. It means that you're trying to constrain the fluctuations of your system. Your phenotype distribution will always stay very close to its optimal, which is E star. That means that if you compare two populations over time, or generally in the stationary state, two populations, they're closer to each other than what you would expect in neutrality. And that's what here you'll see. So the divergence in the stationary state, the sort of the level it levels off to, is much smaller than what you expect in neutrality. And we're building this ratio that the denominator also depends on selection. But what we know from the theory is that the divergence is much more sensitive to selection than diversity. So the equilibration time, so the equilibration time also changes. So this curve saturates much faster than you would expect in neutrality. And that's the time scale that we discussed, is 1/c. To go quickly to a non-equilibrium picture, everything has been so far in a time-independent environment. You were climbing up a fitness peak, and once you get to E star, you're happy. So let's say now that your peak is moving, so your environment changes on much longer time scale, and your population keeps following the environment. Can we formulate that in the same way? So this is probably a more realistic version of evolutionary processes. All right. So let's say this is my picture. This is my fitness so in this axis is now time. This is my phenotype. Your distribution of phenotype is now equilibrating in your own fitness landscape. But what is happening is that the peak of the fitness landscape also fluctuates over time. And that's this green line fluctuation. And population follows the peak. For practical, basically what ends up happening here is that you have the E star that is now time dependent. But let's keep the curvature of fitness function the same, C. You could try to move that as well. We have the first case, we have stabilizing selection, and that's what we discussed before. I didn't use the term. It's called stabilizing selection. It stabilizes your population, so that's C. And then you have environmental driving rate. So let's say your population, your environment is fluctuating also in an OU fashion. So you have a driving rate, and you can formulate it, how your fitness optimum changes over time with some driving rate equal to V, right? Now, if 1/V is much larger than your equilibration time, which was 1/C, right, what ends up being the case is that at each point in time, you have enough time to equilibrate in your own environment. And then next step, your environment changes, and you slowly try to follow the environment. So that's, in physics, you would call it adiabatic process. That's the easiest process to solve in this case. Also, somewhat a more interesting, I would say, dynamics in terms of how you would apply to data. You could solve this equation. These are two coupled Gaussian processes. You can solve it exactly for all regimes, and that's perfectly fine. Maybe it's a good exercise to do. But so let's focus on this one for the moment. What you would end up having, well, these are not both cases. You can write down a joint stochastic process for your population mean gamma and for E star now for your environment, and you write down a continuity equation. So there's a probability current. In the first case, that the environment doesn't change, right? So you sort of equilibrate around your E star. There's a perfect sort of equilibration. There is no probability current after you find your equilibrium. Once you kind of have moving optimum, what you end up having is that you have curls. You have probability current that keeps you going around and finding the new optimum. But it goes around the diagonal, so it's still following your population pretty well. But if you have very fast, rapidly evolving population, rapidly changing environment, your population basically spreads much broadly, right? So it doesn't follow the environment very well. You still have some movement in the population because it's always lagging behind. It's doing things. But it's not very well correlated with what environment is fluctuating. So on average, you get a much broader spread. From sort of practical point of view, now if you compare what the measure we built up before, this divergence to diversity ratio as a function of time, in the absence of environmental fluctuation, we had this saturation level, right? So this conservation curve. Now, if you turn on drive from the environment, you end up having the green curve, which basically says in the small time scales, short time scales that like equilibration time, you basically behave very similar to equilibrating in a time-independent fitness landscape. And then on much longer time scales, you're following the evolution of the environment, and that means your divergence increases because you're moving away from where you were, right? And in this regime, basically, this deviation is linear in time with a slope that is proportional to the driving rate. This is sort of a side remark that the diversity, because it fluctuates very fast, it's insensitive. It doesn't see the fluctuation of the environment. So the statistics of the diversity in this regime are basically not affected by the environment. So this is now a good sort of measure, right? So you can look at the data, plot divergence as a function of time, and ask whether you have drive in the environment or you have a stabilizing selection. And I think I'll have time to actually show one of those plots. But before that, let's try to also conceptually build up one more concept here, and I think that's useful for thinking about adaptation in environments. So I kept telling you that we have, in principle, a non-equilibrium process. So if you were to characterize an equilibrium process, what you end up doing is you build up your Boltzmann distribution, you get a snapshot of your population, and you basically know everything about it. If you have out of equilibrium process, your sort of statistics depend on the history of the population. So what you would need to know is a population history, which is basically, let's call the history now the mean phenotype history, gamma 0 to gamma n, over this time period of t0 to tn. What I'm interested in is how much sort of gain in fitness I had, how much effort as a population I've put in to climb up my fitness peak. So this is an example of the equilibrium. So you have a one fitness peak, and the population starts up down here, and it wants to go up the fitness to the gamma n. And each step, it just climbs up the fitness proportional to the slope at that time. The slope changes, right? So the slope is 0 in the peak. But so it goes faster here and goes slower up there. And you can sort of sum up all these upward arrows and tells you how much total adaptation you had during this time. This is an equilibrium in a sense that it doesn't matter how you go up. Irrespective of that, the total amount of fitness gain is equal to the final fitness minus the initial fitness. So fitness landscape. Now I introduce non-equilibrium, and that means that my fitness landscape is changing over time. I go up here, parallel to my fitness function. I'm up here, happy. All of a sudden, the environment changes, and that means my fitness drops down. I have to climb up again. So you do all this again and again. And what I would say as amount of effort the population has put in is the total sum of the upward arrows again, right? But that's not equal anymore to the end point minus the initial point, and that means that it depends on the history of this evolution. This quantity is called fitness flux, the sum of the upward arrow in both cases. And it relates to a sort of a slightly deeper concept in statistical physics. Again, these are analogies. They are not exact equalities. So it relates to heat dissipation and entropy production in non-equilibrium stat mech. We don't have first law of thermodynamics in a way, right? But we have second law of thermodynamics that fitness flux should be positive. And it's basically equal to the Kullback-Leibler distance. And I think you have heard about these things in information theoretical measures, right? Between now the probability of forward paths over the backward trajectories. Now, instead of looking at probability distributions of states, you look at probability distributions of trajectories, and you build up the relative entropy between the forward trajectories and backward trajectories. And because you're being driven this way, in a forward way, the forward trajectories are way more likely than the backward ones. And this relative entropy basically gives you the logarithm of the ratio of these things, give you the fitness flux. So they're more likely, exponentially more likely, and the exponent is the fitness flux. In the model we built just a couple of minutes ago, the fitness flux, you can calculate what it should be. It's equal to, it relates to the quantities that we built by the divergence curve. It equals to the deviation from the sort of stabilizing selection divided by the level you level off. And I should have made a plot, but let's do it here. So let's say this is my adaptive curve, and this is my stabilizing selection curve. So the omega ad is this guy, and that's omega tab, right? So this was proportional to 1 over c. This curve, this slope was proportional to v, right? Now, at a given time, the further you go in time, the more adaptation you have. So it's cumulative in time. So in the stationary state, basically what you get is the fitness flux is equal to the curvature times the driving rate. So the larger your curvature, the closer you're following the fluctuation of the fitness. So you should have more adaptation. And the faster the environment changes, also the more adaptation you have, because you have to move more. Yeah?
Yeah, sorry, so you use those tools of deviation from behavior. You suggest that you could compare the situation where it relaxes to an optimum with a situation where the environment doesn't change. The situation where this is the case and the environment changes.
Mm-hmm.
But you have these deviations in both cases.
Well,
so how do you distinguish?
So the first case is a transient, right? Yes. So if you were to look at that case in the stationary state, you get fitness flux equals 0 because you have detailed balance. Your forward trajectories and backward trajectories are equal to 0. So that means once you reach the peak, so if you look at the limit of large time, you're in the peak. So you're just fluctuating around the peak, and because you have detailed balance, then your fitness flux is equal to 0.
But do you have a way to distinguish relaxation, a transient in both cases? But
yeah, so you can
distinguishing when the environment changes and when it doesn't.
You can do it by sort of having a very long time trace, right? So basically, how it behaves as a function of time, right?
So sorry, in the DKL formula, when you derive it, you assume that you locally in equilibrium.
Yeah.
Still have the Boltzmann.
You assume that you have microscopic reversibility, exactly. Yeah, so that comes from the mutation.
The Boltzmann formula at each step.
Yeah.
At each transition.
For the mutations, right? So that's what yeah, exactly. Okay. I think I have 10 minutes, right?
Yeah, I mean, you have 10 minutes with questions.
Okay.
8 minutes with questions.
All right, good. So I'll do a very quick example. So I sort of didn't get into half of the talk, but I'll do a quick example of all the things we just built. How can we apply it to a real biological system? And the system I'm going to talk about is gene expression and fruit flies. And you've heard enough about biology to know what gene expression is and what fruit flies are, I suppose. So why is this a quantitative trait? It's encoded, the amount of gene activity or expression level you have is encoded by many different factors in your genome. And that being is the gene regulation, one of the main ones. And that means that there are a lot of transcription factors that bind or unbind from binding sites or promoters or enhancers in your genome. And then they send a signal to your RNA polymerase to transcribe your gene. You make eventually amino acids and fold 4 proteins. And that sort of determines the features of the organisms you deal with, which determine their reproductive success. And then it gets encoded back in the genome. So it's relatively a reasonable quantitative trait to deal with. So let's pick that. And we're looking at 7 species of Drosophila. And they are diverged by about 40 million years of evolution. And we're looking at a sort of finite number of genes in these homologous genes across this species, so about 6,000 genes. And let's try to do a very simple analysis. Let's start from the things that we just built, right? This is not the obvious time trace, right? So what we are basically trying to do is trying to say, let's say my observations are at the endpoints of these nodes, right? So these are the species I see. I have no idea what happened in the past. But I have a way of reconstructing the past in a probabilistic way. And I'm going to sort of trust the model I built a bit too much maybe and try to sort of, under different evolutionary scenarios, I would sum over all possible evolutionary histories that would lead to observations that I have at this time. So the first thing that I want to do is like whether or not I see any evidence for this selection in gene expression levels that I've been talking about. And the answer is yes, we do see that. Let's try to look at gene expression divergence across a species as a function of the divergence time between species. And the time is now measured in units of sequence divergence, and the neutral sequence as neutral as you can get. So these are synonymous mutations among the species that basically mean they don't change the proteins, right? So they're as neutral as you can get. So it's good because then I have some sort of measure of good neutral time on the x-axis. And I measure gene expression divergence, sort of do the proper normalization of that. So I know that if it's neutral, it should saturate to 1. It really does not saturate to 1. What we can see is that this is data averaged over all genes. The error bars are super small, so you put the error bars, you don't even see them. What we are seeing is that at that sort of whole genome level, you average over all genes, you treat them all as different realizations of evolution, and that's not the good thing to do. And next slide, I'll show you that. But overall, we see at the bulk of this distribution, there's evidence for adaptation. It's obviously deviating from equilibration or saturation of the gene expression values as a function of time, and obviously not getting to 1. So we could sort of hand wave away and say there is adaptation going on. We have actually 63% significant adaptation at the gene expression level. So that means that there's response to time-dependent environment across all these species. This is the first step, but we can do better, right? So this was basically a coarse-grained analysis of gene expression divergence. What we can do is try to use the model a bit one step further. So this whole sort of math we built in gave us some probabilistic description of the process. So we can write down probabilities of gamma given environmental states, right? We can do propagators in this case. So you can sort of construct all of these entities from the theory. And you can ask, if I observe values of gene expression here, right, or so between 2 time points, but I don't know what the state of the environment has been. So I can build up this propagator from the theory, and I can sort of trace out the environmental state, your sum over all possible environments that you observe, to calculate the marginal distributions or marginal propagators between 2 different values of phenotypes, right? So this is assuming a model because these propagators now depend on your values of evolutionary parameters, the curvature of fitness landscape, the driving rate, and you get different propagators depending on what parameters you put in. Then once you build this, you can do this along a tree, right? So each branch of, so this is now a 3-species tree rather than 7. Each branch of this tree basically gives you a time separation, and the branching events basically tell me from this point to this point, these 2 species have different history, but from the sort of branching event to the past, they share the history. So you sort of propagate on the tree, taking the topology of the tree a bit too seriously, and estimate the 3 propagators for each gene, right? And then you can sort of revert this and compute the posterior distribution for the parameters of your model. So for each parameters now, instead of driving rate, this is now the composite variable, which is the fitness flux, which is C times V. For some technical reasons, we did this way. So you basically, you want to do what's the probability of the parameters of my model given my observation, and that's you just use Bayesian approach to revert from your sort of estimates here to get this one. And you account also for, you can now, because this is probabilistic, you can account for errors, you can put an error model, how far I am from the true mean of the population and all of this. You package it together, and you get basically for each gene, instead of doing it for the whole organism, for each gene, you get a posterior distribution with sort of maximum likelihood values of driving rate and stabilizing selection. And then you can ask, okay, what genes are more likely to adapt to or to respond to environmental fluctuation, and what genes are basically being more driven to, by the environment, and do some biology sort of analysis there to ask, what are the functional classes that tend to be adaptive. So these are sort of, you can sort of build this reverse machinery, infer selection or fitness landscapes, and then ask more biological questions without being biased towards a certain function and doing experiments in the lab. So this is sort of a slightly more unbiased inference of fitness for gene expression values. And then once you identify genes, you can then go in the lab and do more precise experiments. So this has been sort of a fun application of the theory, which we started building it just for the sake of theory, and then applying it to data, which turned out to be useful, to be honest. All right. I would summarize this talk by basically telling you it's good to build theory, build toy models to first understand how your biological system behaves, and then try to be generic in the theory. I didn't go into details of how insensitive the outcome of the theory was to details of the encoding of the genome. And that's very important, because most times we don't know most of the biological details, and we know them and we are wrong, and that's even worse than the first one. So you really want to have some generic statistics that are insensitive to details. And that's not always possible, but if possible, do that, or try to look for those. And as a result, we managed to look at the Drosophila sort of gene expression evolution without knowing how the genotype of Drosophila maps into these gene expression values. And so that was basically the picture we built. So gene expression is primarily under adaptation, and we identify a lot of functional sort of genes that tend to respond to environmental fluctuations. I would end it here by saying that the next step was, instead of looking at the drive from the environment on a population, instead we were looking at co-evolving populations of pathogens and their adaptive immune system. So this is now a co-evolutionary dynamics with feedback, which basically ended up in looking at adaptive immune system with HIV. So I'm not going into this talk because it's another 20 slides and I'm not talking about it, but it's been a fun theory as well, and sort of now more or less the main focus of my group is on these topics. To give you a bunch of references, I think this is a good place to end. The theoretical part, I think quite a bit of it you can find in these two papers. The gene expression is in this paper, and the thing that I did not talk about is in the co-evolution of immune system and pathogens, chronic pathogens in this paper. With this, I'd like to end and take any questions.